Hello everyone, my name is OrdonezFlores. This summer I worked on a research project titled Visualizing Water Pollution and Quality in New York City using QGIS. This project was completed with Billing Chen, Lauren Rogers, and Dr. Brian Giebel. New York City is a large urban center that has serious impacts on its environment and ecosystem by direct pollution inputs. This summer, I investigated water pollution and quality data from numerous sampling locations as a part of New York City Open Data Portal. Water pollution is derived from many sources. Runoff from land surfaces and direct or indirect discharge of wastewater are also two important sources. Water pollution can lead to significant eutrophication events, harmful algal blooms, hypoxia, fish kills, and overall disruption of ecosystem function. My main goal was to investigate seasonal water quality data for the year of 2019 through February 2020. Our team utilizes GIS, or Geographical Information Systems. Many of you are likely familiar with ArcGIS. I utilize QGIS, a freeware program very similar to ArcGIS. QGIS is a powerful package that allows users to manipulate, process, analyze, and visualize geospatial data in maps. Generating maps require shapefiles or the base geographical layer other data is transposed on. This map contains a shapefile of the shape of the 5 boroughs. Numerical data, including sampling locations in longitude and latitude, measurement values, and other relevant data are saved in CSV files before being imported to QGIS. This map over here shows the value points that represent New York City subway stations. Raster layers are another type of image that add important information to maps. For example, this image of Google Maps gives us a sense of where the points are in terms of the map. In order to help visualize the data, I performed inverse distance weighting interpolations. IDW interpolations are weighted approximation algorithms that mathematically create missing data for unknown points between known data points already in my dataset. We started with New York City Open Data Portal and its New York City Harbor Water Quality Dataset, which included various water criteria around New York City. I filtered and sorted the data for the years 2019 to 2020. After which, seasonal averages were obtained for all criteria of interest, shapefiles were installed, and I processed the data using QGIS to generate the interpolation layers for each season and water quality criteria. I settled on a quantile classification for the interpolation. 2 other options were available based on equal and continuous intervals. As you can see here, the quantile classification allows for a highly resolved gradient with a greater range of colors that produce clear hotspots of accumulated pollution, while the equal interval classification produced a monotone result that was not useful and did not represent the increased values in some locations. Similarly, poor results were obtained for continuous classification interpolation mode. The data I worked with was the surface water measurements, and the shapefile we had was used for the 5 boroughs of New York City. I used the New York City Community District shapefile, which included water boundaries and allowed me to clip the generated interpolation to the Community District layer and superimpose it over or below the 5 borough shapefile. Lastly, I evaluated my interpolation maps and noted any differences or patterns by season. Fecal coliform originates from human and animal waste, which can enter urban watersheds directly from runoff, from wastewater treatment plant outflows, and from combined sewer overflows, particularly during high precipitation events. This is problematic because fecal matter is nutrient-dense and can stimulate algae growth, resulting in eutrophication. Fecal coliform measurements in the interpolation show high values in the summer. Spring, fall, and winter months were nearly identical in having low values in comparison to summer months. High values were observed in northern New York City and the southern tip of Brooklyn. It's very likely that summer fecal coliform values can be explained by a particularly wet summer and illustrates the impact of CSOs. Under dry conditions, sewage waste normally flows towards wastewater treatment plants during the high rain events. Wastewater treatment plants struggle to process water as inputs increase. This leads to CSO outfall by design as seen in the right image. I also evaluated ammonium, a pollutant byproduct of organic matter and detritus decomposition by nitrogen-fixing bacteria. Ammonium values and the interpolation show that transition seasons, spring and fall, have unusually high values in comparison to summer and winter. Jamaica and Flushing Bays appear to accumulate or directly receive high inputs of ammonium. During the fall, increased water circulation contributes to the upwelling of nutrients and promotes algae growth, a phenomenon known as fall bloom. As aerobic nitrogen bacteria decompose these masses of algae, they produce hypoxic conditions. The ammonium produced during this process can be very harmful to the ecosystem, as excess concentrations of ammonium are toxic to many aquatic organisms. As we've seen, QGIS is suitable for evaluating water quality data. There seems to be no seasonal correlation between ammonium and fecal coliform, and this potentially indicates that there may be different sources of each pollutant, or a seasonal time lag between peak seasonal levels. The seasonal lag can potentially be explained since fecal coliform in the summer, being nutrient-rich, can stimulate algae growth and an ammonium spike in the fall when this algae is decomposed. As a next step, we'd like to investigate precipitation rates across seasons, which may account for the high fecal coliform levels in the summer due to combined sewer overflows. In general, I suspect expanding our data would improve our interpolations to more comprehensive data and sampling locations, as seen in this image. This could provide a better interpolation and more resolved gradients. Ultimately, we would like to evaluate chemical inventories, kinetic data, and wastewater treatment plant discharge or magnitudes to model or predict likely locations of new and emerging pollutants. References are included for your use and access. I would like to thank Dr. Tony Kack for aiding our group in using the QGIS program. I'd also like to thank the Alfred P. Sloan Foundation, Advanced Science Research Center, CUNY Office of Research, and the CSURP program for giving us so much support this summer and for allowing many students, like myself, to grow. Thank you.